fungal hyphae. But in mycetoma, we do not know what is the form of what form is mycetoma enter the body. If it is conidia, if it is a mycelia, fragmented mycelia, or it is the grains.
Is it a cutoff point? No role for toll-like receptors?
You cannot say there are no rule for toll-like receptors in mycetoma infections because until now we do not know what is the immune response of mycetoma infections, because from all patients of mycetoma infections, there are one type of reactions. Type 1 reactions, mycetoma grains surround for either to be mycetoma or actinomycetoma surrounded by neutrophils. Neutrophils, it is not specific cells.
So you can consider it in your PhD program, because I like this type of work, because it's recent, as you know. It's not, so you can include it in your PhD.
For me? For me, the other lines of the study to do this is study in mice.
I think immunological study in mycetoma, as we said till now, not fully done yet. We need to rule out many cells if they had a role in mycetoma or not. Toll-like receptor or macrophage, role of macrophage in mycetoma actually not fully worked. Concerning the question about FICOL, first day I took my samples for Institute of Endemic Disease, and so my friends they do magic like, and they separate the white cells I did not find any problems. Even I'm newly trained in this.
Did you test the recent one?
No. Okay. Thank you.
There is another question over there. Yes?
Thank you for the opportunity. Dr. Hamad Osman, Registrar General CRA. After confirmation of the increased level of interleukin 10, the pathogenesis of mycetoma, can we depend on interleukin 10 level to determine or detect patient with recurrent of Madura after response to treatment, either surgical or medical? Can we take it as a marker for that or not? Thank you.
Okay. For this marker or for this, say we need another cohort study or prospective study in cohort study in the field for detection of the early infection. We need to follow the cases to detect that is it good or not, and this need high fund.
Thank you for your talk. In fact, this is an extraordinary disease because it is caused by fungal and bacteria at the same time. Have you ever isolated actinomycetes from these patients? And if so, is the immunological response the same as in Madurella or it is different?
The response in Madurella is different. We didn't study the response in actinomycetes. Just we studied our study was in Madurella mycetomas.
No isolation for patients.
From our patients, no isolation, only just Madurella mycetomas.
Good morning. My name is Dr. Sarah Ahmed Tijani. I'm a registrar in clinical microbiology and infectious diseases at the Sudanese Medical Specialization Board. During your talk about your study or your master, you were mentioning that ketoconazole responders and non-responders patients. Okay. On what criteria was based responders and not responders? And did you take in your study the advantage of the length of treatment? Because it's very important whether the patient is taking ketoconazole for a long time or not. Thank you very much.
You mean links between the ketoconazole and cytokines?
You said responders
Yes.
and non-responders in your study.
Yes. Yes.
So according to what did you mention responders or non-responders? Is it clinically, laboratory-wise, or what? And the second thing, the length of the treatment.
Okay.
Thank you.
Okay. All the patient, at least we took 6 months after treatment. All the patient, for all the patient. And then we measure, we took the samples. Even patient more than 6 months, more than 1 year, they did not respond. Every patient did not respond more than 6 months is included in our, we call it non-responders. And the other are responders. Also they used to respond more than 6 months. Response also more than 6 months or less than 6 months is the responder. Response clinical, no laboratory, yes. Closure of the sinus, regression of the lesions, relief of disability, and so on. No, we did not do that.
I think now we can go to our third speaker, and maybe after that we can have more discussions. Our third speaker is Dr. Paul Hoskisson, and he's a lecturer in microbiology, University of Strathclyde in Glasgow. And his primary area of research is actinomycetic biology. And he will tell us about the genetics of the actinomycetes.
Right, okay, I'd like to thank the organisers for inviting me. It's been a really interesting few days, not only to be in Sudan, but also today we've been to the clinic to meet the patients. So it's really brought home to me the importance of the work that we're doing. Obviously, as a biologist, I tend to look at this problem from the aspect of the organism and working at the molecular level within the organism. So I think with the microphone, I feel like I should be doing karaoke. So it's kind of a bit difficult. So remind me if I drop the mic down from my face. So my talk is kind of a bit of a shift from what we've seen so far, and I'm going to talk about the work we've been doing in my lab in Glasgow on trying to develop tools to study mycetoma causing organisms, both from an aspect of their biology, but also to try and type these organisms and try and understand how they've evolved, how they evolve in patients, and which strains are causing which kinds of disease. So this picture here, this is the new building that we've just moved into. It's a £35 million biomedical research centre. So my lab is up here on the top floor. So we have lovely views like this over Scotland. No, I'm joking. This is actually out in the islands, and this is my research group currently at the moment. Selena's working on mathematically modelling growth of Streptomyces, and Laura's working on conserved virulence factors within Streptomyces. There's just another view of our building. So the outline of my talk is I'm going to give you a brief introduction, which I'll largely skip through because we've had such a good couple of days introducing mycetoma. I kind of don't feel necessarily that qualified to talk about mycetoma anyway. And then I'm going to talk about the work we've done with Nocardia, about multilocus sequence typing systems that we've begun to develop with Nocardia. And really, whilst Nocardia is not a primary mycetoma causing organism, certainly in Africa, I think what this does emphasize is the fact that we can take these techniques and start to apply them to Streptomyces somaliensis, Streptomyces sudanensis, and potentially the new strains that are going to be named later this year. I'm going to talk about some of the genetic tools we've been developing for Nocardia, so we can start to look at the function of virulence genes and try and really understand what's going on at the molecular level with these organisms and why they're causing these kinds of diseases. I'm going to give a quick interlude into some work that we've been doing on a search for new drugs against Nocardia, but the screens again are applicable to lots of different mycetoma causing organisms. And then the last part of the talk, I'm going to round up with some really exciting news about the whole genome sequence of Streptomyces somaliensis. So I think this gives us a really good opportunity to really dissect this organism out at the molecular level and really understand why it is that this organism can cause these kinds of debilitating diseases. And then hopefully I'll try and explain to you how we can learn from comparative genomics the functions of these organisms. So, as we've heard, actinomycetoma is a severe debilitating infection, can spread to deep tissue with bone implications as well, characterized by these formation of large masses. In terms of actinomycetoma, Streptomyces, the Nocardias, and the Actinomajoras are the three important genera. But as we've seen from the last few days, we need better diagnosis, more rapid diagnosis, so we can begin treatment much more rapidly. A better understanding of the biology, so we can then target therapies to better combat these organisms that are responsible for this condition. So again, as we've heard, there's no really accurate figures in rural Africa, the Middle East, near tropics for this condition, and largely treatment appears to be kind of random in terms of, oh, we'll try this antibiotic and see whether we get a response from the patient. And often the results are mixed. And again, this harks back to the fact that we don't really understand the phylogeny of the organisms that we're working on that are causing these diseases. So if we can add better resolution to our understanding of these organisms, then we can perhaps target therapy much more readily. Often surgical interventions often required, and we heard from Ahmed the other day about the socio-economic problems that this involves and the implications for the sufferers. So in my eyes, as coming at this from the position of the organism, there are several questions that we have regarding these organisms and the fact that they cause these mycetomas. We know little of their phylogeography, which organisms are present where, and how their relationship to each other is, and how they cause these diseases. Lift it up, sorry, karaoke mistake. So which strains cause the most disease, certainly within the actinomycetes, and where they're found, where they're prevalent? Do these different strains predominate in specific manifestations? So do we see a specific strain of Streptomyces somaliensis that's likely to have bone involvement, or just at the cutaneous level? So we really need to understand this. Is there an ecological or a geographical limit to the strains? Do we only find specific substrains within a specific area within Sudan or within Mexico? And Oliveira alluded to this the other day with the separation of Nocardia brasiliensis into several strains. And if these geographical variants are different, why is that the case? And I think getting down to the nitty-gritty of what's going on with these strains is very important. Another interesting thing about these organisms is we know so little about the genetic characteristics and the proteins on the surfaces of these organisms that really interact with the host, and the virulence mechanisms that they use to invade and persist within the host and replicate. And an area that kind of goes back to my early work that I've always worked on growth and development in actinomycetes is I believe that morphology is probably important in these organisms because they're hyphal and they grow apically from the tip. This probably gives them the benefit in terms of being able to invade tissue and invade bone in order to carry on with the infection. And one of the ways that we can approach this is to look at their whole genomes, and also that we can glean a lot of information from the genome structure of these organisms. What are the common virulence factors between the organisms that cause mycetoma, and maybe from this we can lead to targeting drugs specifically to specific targets that these organisms have. So, as I'm sure you're all aware, in Nocardia it's a filamentous fragmenting. We used to think it was largely a saprophytic actinomycete, and I think probably this is the case, but we see more and more pathogenic Nocardia isolated, certainly over the last 10 to 15 years. By bacterial standards, they have quite a large genome, about 6 megabases, so 6 million bases within their DNA. There is one fully sequenced strain that's Nocardia farcinica, But interestingly, from an actinomycetoma point of view, this was an isolate from the bronchus of a 68-year-old man in Japan. So the likelihood of being able to infer a great deal from this genome in terms of what are the factors for causing mycetoma, maybe it's not particularly valuable to us. And I think sequencing strains that are directly from mycetomas will be very important. As a single circular chromosome, and as characteristic with a lot of actinomycetes, it has a very high GC content within its DNA, and the genome encodes about 5,674 genes, give or take the odd pseudogene that's likely to be found within that list. So this gives us a lot of scope for these organisms in terms of genes that are likely to be involved in virulence and growth within the host. Another interesting aspect of Nocardia biology is if you look at the phylogenetic tree of these organisms, I'll show that on the next slide, you tend to see the pathogenic organisms spread throughout the lineage so they don't cluster together, which I think is important for trying to answer our phylogeographic point of view. So there's currently about 75 species recognized, and there's a lot of diversity in terms of morphology and the appearance of these organisms on plates. Some of them produce abundant pigment, some of them don't produce any pigment at all. A lot of these strains don't produce any aerial hyphae, but within even a short range of strains that we've studied in my lab, there is a lot of variation. So this suggests that there's a lot of plasticity within their genome. And whilst we see this phenotypic variation on plates, if you look at them at the 16S level, the rRNA diversity within the Nocardia is relatively small, and fatty acid discrimination is relatively poor. So we need to develop methods to try and put a little bit more resolution onto these trees and try and understand what's going on. Okay, so how can we improve our knowledge of typing of these strains? How can we differentiate between strains that are isolated from mycetomas and from patients, and try and add value to this analysis to try and develop a typing scheme? So traditionally people have used things like PFGE, which is pulse field gel electrophoresis, arbitrary primed PCRs, multilocus enzyme enzymology, electrophoresis, which is frequently used to look at global epidemiology of these strains, where you can differentiate between very closely related organisms based on characteristics of their DNA and also of their protein. But what these rely on is largely random parts of the genome that are uncharacterized, and analysis of these is often difficult, and interpretation in terms of the overall biology of the organism is very difficult. So, excuse me, one of the ways we've been trying to address this in Nocardia, and this scheme can easily be rolled out into many other organisms that cause actinomycetoma, is using a multi-sequence, multilocus sequence typing system. And this applies nucleotide sequencing to known genes, so you have an idea of how likely these genes are to evolve rapidly within the host, or to be relatively free from accumulating mutations, because we tend to use housekeeping genes. So this scheme was developed by Brian Spratt in Imperial College to look at typing lots of different pathogens. So you choose 7 housekeeping genes, so these are genes that are required for normal function within the organism, and you amplify about a 500 base pair fragment of the gene. And then you can sequence this, and you can look for specific alleles within each sequence, so single base pair changes or inversions or deletions within those sequences, and you can assign a number to each of these alleles. And then for somewhere like Sudan, it's ideal that you can then type your strains. Sorry, I'm talking too long and it went to sleep. You can then type your strains via the internet, so it allows you to use a global resource via the internet to then type your strain and add it to a database of strains, so then immediately you can see what kinds of strains have been isolated that are very close to this strain, very distant. And this is in its early days for us, but I think with a lot more strains we can begin to develop this. So there's a schematic for how MLST works. You make a chromosomal DNA prep of your organism, you can see here, and then you amplify your 500 base pair fragments of 7 housekeeping genes, and you sequence these strains in both directions to ensure that you're not just picking up single mutation in the sequencing. You can compare these sequences of each of these known alleles with a genome sequence to make sure that you're amplifying the right region. And then you can assign the alleles, as I said earlier, to give yourself a profile for a specific organism. And then you can compare and you can cluster each of these profiles, and then you can begin to link this to the clinical data. And I think this will add a lot of resolution to what we know of these organisms currently. So we set out to develop this system in Nocardia. So we went to the MLST database and we took all of the gram-positive organisms that have been studied by this system and looked for housekeeping genes that have been used in these systems. And we identified 7 or 8 genes that have been used in all of the gram-positive organisms, and some extra ones that have been used in more closely related organisms. And then we designed PCR primers after we checked that these genes were present in the Varsinica genome. And we used a program called Gene Fisher, which is available freely on the internet. So again, it's very easy to do this. You don't need complicated software or expensive software to be able to do this. The 7 genes that we were able to find encode various housekeeping functions within the organism, and we were able to show that we could PCR up all of these genes from our strains, and then we could then begin to look at sequencing these fragments to then learn more about the sequences. And rather than go into great detail about this, I've just focused on one strain. But what this allows you to do is see very minor variations within the sequences. So this is a range of Nocardia Varsinica strains, human and animal isolates, and also a couple of, when the pointer comes on, a couple of strains like Nocardia abscessus and this is the Varsinica sequence strain. So what you can see is that whilst the sequence is very highly conserved, you can pick up single mutations which allow you to make almost a fingerprint for your organism, which then allows you to be able to look at how these strains have evolved. Okay, I'm not going to go into great detail of these kinds of changes. All we need to think about is that most of these changes are largely don't change the amino acid sequence of the protein that's encoded for by this gene. So they're largely silent DNA changes, so they really do give us a good fingerprint for understanding what's going on with these organisms. So then for each of these genes you can build a tree, and the trees have highly variable topology based on each sequence. So the way around this with multilocus sequence typing is that you can concatenate these sequences, so you join all of the sequences together to make one super gene, if you like, that you can then build a phylogenetic tree from. And this is just a small subset of the strains that we've done so far. But I think it illustrates our point very nicely of the power of this kind of technique. This is just examining some Varsinica strains. The resolution of the phylogenetic tree is increased. What we have, and it leads on from yesterday's discussion about the involvement of animal reservoirs, is that we see different strains present in goat mastitis than we do in human strains. And also these are some strains that Mohammed Hamid, who's known to some of you, isolated from milk and also from goats when he was working with Mike. But interestingly, the 3 human isolates, sorry, I'll go backwards, are highlighted here. But what we see is this strain here, it's actually a West African isolate, clades quite nicely with some goat mastitis cases from Sudan. So I think what we're looking at is an organism that's broadly spread across the Mycetoma belt, but it does cause disease in humans and in animals. So maybe our animal reservoir is not too far away. So I think what we need to do with these fingerprints is add more strains to this. And this is what we're trying to do now, is try and amplify these sequences from lots of different clinical strains. So any people who isolate Nocardias in the Sudan and around the world from our other international visitors, I would be quite happy to add these strains into this database, and we can slowly build up and get a picture of how these different types of organisms are causing different kinds of diseases. So I was kind of interested and also disappointed that a paper came out at the end of December last year, where this group in the US had started to apply multilocus sequencing typing to Nocardia, but they've done their strains just with 4 genes, which means that it won't fit into the MLST.net database. So I think our system still has a lot of utility and can add that level of resolution. So we need to really look at a range of strains, a range of geographical areas, different disease manifestations, human, animal, and environmental isolates to really add some resolution. And then this tool that you can see here is a program called eBurst. So when we've done a lot of strains, we can then look at how these strains have evolved. And I can get the... So to interpret these diagrams, what you have is a lot of strains in the center, and then isolates that have picked up single mutations act as satellites, and then these satellites that lead again to the evolution of novel strains bud out as different nodes within these graphs. And what would be interesting to do with these strains is also to look at the reoccurrence of mycetoma over a period of time within a host. So we've had a lot, and we've seen a lot of patients this morning where they've had a mycetoma. They've been treated either medically or surgically. The mycetoma has gone away for a while, and then it's come back at a different site. And I think using these kinds of techniques to look at how the strains have evolved within the host and how they may have been dormant within the host for a period of time and then resurged is very important to try and understand these long-term aspects of the disease. I think also we can add, when we know more about virulence factors, we can then do this VMLST, where you then add virulence genes to your analysis as well. So you increase the number of genes in your typing situation, and then you can start to add even more resolution to these organisms. So in these organisms, there are very few detailed studies on virulence mechanisms. There was some nice work has been done by Jun Ishikawa in Japan on Nocardia isolates, and they showed that RPOB, which encodes one of the subunits of RNA polymerase in Nocardia, is actually duplicated within the genome, and this has implications for rifampicin resistance and any other genes that target any other drugs that target RNA synthesis. So by understanding the fine genomic level of these changes within the organisms, we can really get to grips with why strains don't respond to specific drugs. A recent study on nocardobactin is also very important because these siderophore molecules are important for accessing iron within the host, which is an important nutrient for a lot of organisms when they're grown within the host, which is surprisingly often limited to the organism when they're in the host. So organisms invest a lot of energy in trying to get hold of nutrients from their host. The mycelated cell walls in Nocardia are also likely to be very important virulence factors for these organisms. They offer a barrier to getting drugs into these organisms, but also in related mycolate-containing organisms such as tuberculosis, they've often been shown to be important for immunomodulation and how the host responds in terms of fibrous tissue formation, etc. But despite these organisms being known as pathogens for a long time, we know very little about the mechanisms that they use. And as I said earlier, how they invade, they replicate and persist in hosts. So if you look at the Nocardia genome, you can predict some genes that are known in other organisms likely to be involved in pathogenicity. There's these MCE gene clusters which are amplified in Nocardia, and they're known to be involved in mammalian cell entry, hence MCE, in mycobacterium that are involved in these organisms being able to utilize cholesterol and other sterols within the body as a carbon source. Free glucose and free carbon sources are very difficult for these organisms to come by in the body due to various mechanisms. So being able to utilize things such as cholesterol, which are abundant in eukaryotic cell walls, offer these organisms an advantage to be able to live within the host. I mentioned iron acquisition. The organism has 4 copies of the catalase gene within its genome, which suggests to me that it's likely to be constantly combating oxidative stress from the host, whether that be via the immune system or via general growth situations. So again, these are often offering a selective advantage within the host. Intrinsic antibiotic resistance is a big problem with Nocardia, and the sequence strain Nocardia farcinica was shown to be intrinsically resistant to 28 clinically used antibiotics. So obviously this will affect our ability to choose frontline treatments for these diseases, and we know very little about the clinical strains that were isolated here in terms of their antibiotic resistance on a broad scale. I've put a production of immunosuppressants, so it's well known from Nocardia brasiliensis that they produce a molecule called brasiliacardin, which has been shown to have immunosuppressive effects. And as we know, actinomycetes are well-known producers of secondary metabolites. So if these organisms are producing molecules whilst they're in the body, they're actually reducing the efficacy of the host immune system, then maybe that's how they start to get hold of the host body and be able to replicate within them. But I think to really dissect this out, we need more experimental work to try and cover this. So one of the things we've been doing in my lab is trying to develop some genetic tools for Nocardia. And obviously with this high level of antibiotic resistance, developing selectable markers within these strains to look at gene knockouts has been very difficult. So what we've done is we've taken an approach where we've used established techniques from Streptomyces, and we've looked at vectors that contain resistance markers for apomycin, neomycin, thiastrepton, hygromycin, which are not particularly useful drugs clinically, but they, neomycin aside, obviously, before any of the medics take me to task. But we can then use these antibiotics. If we can determine the antibiotic sensitivity of these organisms, we can then use these selectable markers. We've been optimizing techniques to introduce DNA into Nocardia so we can then develop these genetic techniques and develop plasmids and gene knockout systems to study gene function. And one of the interesting ones we've been looking at is this integrating vector, PSET152, which carries the napromycin resistance gene. And as you can see, this intrinsic antibiotic resistance limits our marker use. But apromycin, 100 micrograms per mil, is a very high level of antibiotic resistance, but it allows us to use these vectors, and there's a selection of other vectors there, and their utility in terms of studying gene function for integration, for mutagenesis, shuttle vectors, which allow us to move DNA between E. coli and Nocardia, which speeds up our cloning techniques, and also to look at localizing these organisms by expressing green fluorescent protein, GFP, within the cells, so we can actually localize these organisms in experimental animal models. So I think we're on the verge of being able to do some really interesting work with these organisms. And I put this slide in just to show that we took a range of Nocardia strains and tried to introduce these vectors. And what you can see just by a simple plus-minus test is actually we see a range of resistance in these organisms to both neomycin and to apromycin. So Nocardia farcinica and brasiliensis show much more antibiotic resistance to these two antibiotics than say abscessus and gamkensis, and some of these are environmental isolates, so we're trying to look at a range of strains. So one of the vectors that we've looked at, and I think this is interesting in terms of the soil ecology of these organisms, is PSET152. It's a conjugative integrating vector, so we can conjugate it from E. coli via bacterial sex into Nocardia using standard Streptomyces protocols. And it integrates in Streptomyces the attachment site for a bacteriophage Phyce31. So this bacteriophage does not infect Nocardia, so the obvious expectation is, well, why would we use this vector in Nocardia? But we tried introducing this vector, and it integrates, and it's stable within Nocardia, so it must integrate. So we looked through the genome of Nocardia faucinica for pseudo-ATB sites, so the attachment site for the phage within the genome, and we were able to find several potential sites where this vector could integrate, and we've shown by southern blot, which I'm not going to go into great detail, but this vector is stable within the genome, which means that this bacteriophage must be or must have a similar relative that's in the soil with these organisms that's able to infect Nocardia. And probably a lot of you are thinking, why is he going on about bacteriophages? But these are really important drivers of bacterial evolution. These are often the ways that antibiotic resistance and virulence factors are moved amongst organisms within the soil and within the environment. So having a bacteriophage attachment site that's common to Streptomyces and also common to Nocardia could potentially suggest to me that there is genetic exchange between Nocardia and Streptomyces, and of course we know that both of these organisms are important causes of Mycetoma. So in terms of developing our genetic knockouts, we're hoping to target these MC genes, some essential sensing systems within the organism to try and dissect out the biology. We know that the MTRAB system is very important in other pathogenic actinomycetes like Corynebacterium and Mycobacterium in causing disease. We'd also like to look at disrupting mycolic acid biosynthesis in Nocardia to see whether this has an immune modulatory effect in animal models, and also look at the cell biology of these organisms to see where and how they're causing disease, whether the hyphae are penetrating between the cells within the infection. So just a quick skip through an application that we've been doing with some of our collaborators in Strathclyde. They were interested in a talk that I gave on Mycetoma, and they offered their services to screen their library of natural products, semi-synthetic derivatives of natural products, and completely synthetic drug-like molecules that have been developed over the last 30 years in Strathclyde. Currently, we've screened about 7,000 compounds against Nocardia faucinica, but we're hoping to extend this to Streptomyces somaliensis very soon. What we've been able to find is a few compounds that potentially have novel antibacterial activity against these organisms. So we use a very, very simple Alamar blue assay. So where you see the blue colour in this 96 well plate, there's no growth of the organism, so the compound has killed the organism. Where you see the pink colour, it means the bacteria has grown and reduced the Alamar blue within the assay and turned it to a pink colour. So this allows us to screen rapidly lots of different compounds. So as I said, we've screened about 7,000 compounds so far, and we've isolated several potentially useful compounds. And this assay is also amenable to MIC determination, so we can develop that very quickly. And we can look at the levels of MIC within these organisms, and this is just a range to show that often these compounds show activity against Nocardia and Mycobacterium, but not against Staph aureus and E. coli. And I think really that the future of drug research is maybe looking not for broad spectrum drugs, but actually targeting drugs to